Hello, I'm Joe Takahashi at the University of Texas Southwestern and the Howard Hughes Medical Institute. And for this second lecture, what I'd like to do is to give you 3 examples of using genetics in the mouse to really discover genes that are involved in controlling circadian behavior. And so the way a genetic screen in a mouse is performed is to use a chemical mutagen such as ENU, or ethyl nitrosourea, which is used to treat a male mouse. That causes mutations in the germline of that mouse. And then that treated mouse is crossed to normal female mice in what we call the G0 generation. And these mice then produce a number of offspring that we call G1 or generation 1 mice. These mice are carrying mutations that they inherit from this treated mouse up here. In a recessive screen, which is a little bit complicated because it requires 3 more generations of crossing, a male mouse is then crossed to another normal female mouse to produce a second generation of progeny. And then the female progeny in this second generation, or G2 generation, are actually crossed back to the G1 male. And that produces the third generation of mice, which are called G3s. And the reason we do this is to make any mutation that was heterozygous or single copy in the G1 mouse homozygous. And in this particular procedure, we try to screen about 20 G3 progeny for each G1 mouse. That gives us about an 85% chance of detecting a homozygous mutant mouse in the G3 generation. So in this kind of screen, shown here where we use circadian rhythms, this particular screen we screened over 3,200 mice. And this histogram here shows you the distribution of period values, similar to what I discussed in the first lecture, for these 3,000 mice. What's remarkable about this behavior is how similar each of the mice are to each other. So the average in this experiment was about 23.2 hours for the whole population of 3,000 mice, but the variation within that population was only 0.2 of an hour. That's only 12 minutes between mice. And so in this screen, this is, again, an activity record of a wild type or typically normal mouse, first on a light cycle, then transferred to darkness so we can see its circadian period. We found this mouse, shown in the middle panel, which has a 26-hour clock. And this mutant we named Overtime. It turned out that this mouse didn't breed, but we were able to recover the mutation from a SIB, this mouse below, which turned out to be a heterozygous carrier. And using this mouse, we were able to map the mutation to this region of the genome. And once we were able to reduce this interval, we found that there was only a single mutation in this gene here, which is called FBXL3, which is an F-box protein. Now, an F-box protein turns out to be involved in a complex of proteins that are involved in modifying other proteins with ubiquitin chains. And these ubiquitin chains then target that protein for degradation in the proteasome. So to make a long story short, FBXL3 uncovered a new F-box protein that targeted the cryptochrome protein for ubiquitination, and that mark then tags cryptochrome for degradation. And in the absence of this enzyme, or loss of function of FBXL3, the degradation of cryptochrome protein is slowed down tremendously. And what that does is to prolong the circadian period and lengthen the period from 24 to 26 hours. So we can see that this particular step accounts for a few hours out of 24 in the feedback circle. So in a second screen shown here using a slightly different strain of mouse, so the period of the rhythm is a little longer. It's 23.7 hours in this case. We found a short mutant, and that's shown here. It's only subtly short, about a half an hour short. Named that mutation pastime. And when we mapped this gene, it turned out to be in a new position. And incredibly, this gene turned out to be another F-box protein. This time, it was called FBXL21, which is called the paralog of FBXL3. So a paralog is a gene that has a very similar sequence that arose from gene duplication in the same organism. And so we assumed that FBXL21 should have a related function to FBXL3. But the mutant is surprising because it has an opposite phenotype. Instead of lengthening the period, as we saw for overtime, this mutant shortens. And so we looked for the interaction of overtime, the long period mutant, with pastime, the short period mutant, by crossing those two strains of mice. And the bottom shows the double mutant, and these histograms show the average period or the period distribution of these four genotypes of mice: wild type, overtime mutant, which are 26 hours long, pastime mutants, which are half an hour short, and then the double mutant, overtime and pastime. And what you can see is that the pastime mutant actually neutralizes the period lengthening effect of overtime and normalizes the period. So this kind of genetic interaction is non-additive and is interesting because it suggested that pastime was somehow counterbalancing the effect of overtime. Now, I'm not going to go into all the details, but I'll just jump to the end of this story, and that was that we found, to our surprise, that FBXL3 is really only found in the nucleus, but FBXL21 is found in both the nucleus and in the cytoplasm. And that in the nucleus, there's a very fine balance between FBXL3 and 21 interaction for the cryptochrome protein. And what happens is that FBXL3 is normally promoting the degradation of cryptochrome, but 21 actually competes for that and protects cryptochrome from degradation. And so that's why we think the two genes have opposite effects. 21 protects cry. FBXL3 promotes cry degradation in the nucleus. So they're counterbalancing there. But the surprise is in the cytoplasm, FBXL3 still degrades cry, but it does more weakly than FBXL21 still degrades cry, but it does so more weakly than FBXL3. So the discovery of these two mutations in related F-box proteins has, first of all, told us that the nucleus is very important in controlling the degradation of cry, and that the primary effect of these mutations on period length appears to act at the level of the nucleus, not the cytoplasm. So that was a little bit surprising. And the second is that related F-box proteins might have very different functions in the cell, both location, but also on their effects on the clock system even though they look very similar at the sequence level. Okay, so this has really led to a revision of our clock gene network shown here. This is the core clock BMAL per cry feedback loop. And now we've modified the diagram so that FBXL21 is really the primary E3 ligase in the cytoplasm. There is no FBXL3 as we thought before in the cytoplasm. Instead, most of the action is actually occurring in the nucleus, where FBXL21 and 3 are competing for cry in a very fine balance to regulate the stability of the cryptochrome protein. Okay, so what I'd like to do now for the third example is to go back to the clock gene and to tell you about another kind of genetic interaction that we've discovered that involves a much more general background effect of mouse strains on the phenotype of circadian rhythms. So a number of years ago, we looked at the phenotype of different inbred strains, and this is just giving you an example of two inbred strains, C57 Black 6J, which is the very common inbred strain that we use, has a beautiful circadian rhythm. Two records are shown here for you, a record from a female on the left and a male mouse on the right. Both of them have very precise, robust rhythms that you can just see visually. And then on the bottom are two records from another inbred strain called Valb-C. And this mouse is very commonly used in the laboratory, but as you can see, its circadian behavior is not so clear-cut as Black 6. The pattern is much more fragmented, the period is shorter, and the rhythm is much less stable. And so using these two strains, we can cross the strains and then look for genetic features that are inherited from these two strains to affect circadian phenotypes. And again, I'm not going to take you through the details, but in this analysis of these two strains, we found about 14 different regions of the genome that are responsible for controlling different phenotypic aspects of the rhythm between these two strains, their period, their amplitude, how robust they are, things like that. And these are all indicated in these oval circles with different colors. And what you can see is they're scattered across the genome. And what's interesting is their locations are essentially in regions of the genome that don't contain known clock genes. There's only one case where there's an overlap between a known clock gene, casein kinase 1 epsilon on chromosome 15, and one of these quantitative trait loci or QTLs for rhythms. All the rest of them defined new genes. So what does that tell us? That tells us that even though we have this clock gene network with a core set of genes, there's still many additional genes that can affect different parameters of the circadian system and that we don't really know about at the molecular level. So the final part of my talk, I'm going to focus on trying to identify one of these types of quantitative genes in the mouse genome using a very specific case. So when we are mapping the clock mutation, we use different strains of mice to cross the clock mutation with in order to have genetic markers that we can follow in these crosses. And in one of the crosses that we made, we crossed the clock mutant mouse to the Valb-C mouse that I just discussed. So what we have here are records from wild type mice or clock heterozygous mice, but they're in different genetic backgrounds. The top two are from C57 black 6 mice, and you can see what I showed you before. The clock mutation lengthens the period by about 1 hour. Okay? But when we cross to BALB-C, even though this mouse on the right is a clock mutant, its period looks normal. So we call this genetic suppression. The BALB-C background is somehow suppressing the period lengthening effect of the clock mutation. And if we were to make 4 additional crosses to BALB-C, shown here, we get complete suppression of the clock mutant. And this is shown here on this graph on the right. In the black 6 background, we see this period difference. In the F1, we see suppression of about 50%. And then in the 4-generation cross to BALB-C, we can see complete suppression of period. So that's at the behavioral level. This suppression also occurs at the tissue level. So shown down here are per luciferase recordings of the suprachiasmatic nucleus and the pituitary gland. And we see that even at the tissue level, there is suppression of the period, lengthening of clock in these 2 tissues. So this is more of a global effect, not just behavior. So how do we find this suppressor gene? So we can approach this by trying to genetically map the suppressor. And this is done by comparing different types of crosses. So here's the clock mutation on a pure B6 or C57 black 6 background. This is the F1 that we saw before. And then these are 2 crosses that are called back crosses, back to either black 6 or BALB-C, shown in green. And then this lower panel is an F2 cross, which is a cross of 2 F1 mice of this type here. Okay? And what you can see is the period distribution is much broader in the BALB-C N2 and this F2 generation. And this is really, we think, due to this genetic diversity. And so what we can do is we can take these F2 mice and type each of the mice, because each mouse is a unique combination of black 6 and BALB-C DNA in their genome. And we can scan each of their genomes with DNA markers to type what their genotype is across the genome, and then see if there's any particular kind of association between a BALB-C piece of DNA and this period suppression of clock. And so that's what's done here in this graph, which is scanning the genome with different markers. Each of these numbers represents a mouse chromosome from 1 to 19, and then the X chromosome here. And what this shows you is that on chromosome 1, we find a peak, highly significant, which shows an association with suppression on chromosome 1, but really nowhere else in the whole genome. Okay? At the bottom, if we blow up chromosome 1, which is the largest mouse chromosome, it's 200 megabases in size. The red line shows this initial peak. It's very broad. It covers almost half the chromosome, which is 100 megabases. It's impossible to find the gene in such a big region. And then in blue and green are shown additional crosses that try to isolate this region of DNA for many generations, up to 8 generations of crossing. And what we see is that the distal tip of chromosome 1 now is the region that carries the suppressor. Okay? But unfortunately, this region is still huge. And so it's really difficult to find a gene in such a big region. But to prove that that region of chromosome 1 really carries the suppressor, what we do is we isolate that piece of DNA on a black 6 background. So only the BALB-C DNA on chromosome 1, this segment shown in green here, and this representation of chromosome 1, which is now 25 megabases, this piece of BALB-C DNA by itself can suppress the clock mutation, as shown in these activity records here and in this graph here, the red line, which is showing suppression. So this tells us the BALB-C suppressor of clock is within this region because we've completely isolated it on that piece of DNA. But again, there are way too many genes to find the gene in 25 megabases. So how do we go on from this point? It turned out in 2002 the mouse genome was sequenced. And one of the interesting discoveries in the sequencing of the mouse genome was that when we look at different inbred strains of mice, we find different blocks within the genome that are either similar or very different to C57 black 6. And that's indicated in these bars here, where this is a comparison of the strain 129 with black 6. Red shows the differences, and light blue show the similarities in those two strains. This is another strain comparison, C3H with black 6, and then this is BALB-C with B6, the comparison we've been talking about. And what you can see is that there are little blocks of DNA that are interspersed throughout the genome. And so we now know from more recent complete sequencing information that classic laboratory inbred strains of mouse are really hybrids of many different kinds of mice, primarily 3 wild species of mice: Mus domesticus, shown in blue, Mus castaneus, shown in green, and Mus musculus, shown in red. And in general, the laboratory mice that we use today are really derived from mouse breeders that domesticated mice both in Asia and in Europe. And these mice were ultimately derived from these different natural wild progenitor strains. So inbred strains are really not pure species. They're really mixtures of different species of mice. And so this led to the idea that perhaps the suppressor of clock might be an ancestral allele that was inherited from one of these different species that contributed to inbred mice. And so to test this idea, what we decided to do was to cross the mouse clock mutant to different inbred strains of mice, shown here, and ask, does a different strain of mouse suppress clock or not? Okay. And the data would look like this. This is a suppressed mouse, and this is a non-suppressed mouse. So Shimamura, who did this experiment, crossed to 15 additional strains of mice, and he was able to find that 8 strains carried the suppressor for mice, shown at the top here, and 7 additional strains, in addition to C57 black 6, failed to suppress. These strains are shown on the bottom here. You can see the red lines are horizontal or parallel. Okay? So this experiment confirmed our hypothesis that it was likely that the suppressor in the genetic background of BALB-C was an ancestral allele that was carried by many other inbred strains of mice. And so we could then take advantage of that by looking within the 25 megabase suppressor region of chromosome 1 and look at all the sequence differences in these 15 additional strains of mice in comparison to black 6. So these histograms show all the sequence differences with black 6. The top strains shown in green are the suppressor strains, and the bottom strains shown in blue are the non-suppressor strains. And within this 30 megabase region, we could find only one very small interval shown here in green that matched perfectly the pattern of suppression and non-suppression among these strains. And so that suggested that the suppressor was restricted to this narrow region, which is about 900 megabases in size. And when we look at this region here blown up, we see that there are actually 22 genes there. And so 22 genes is actually a number that we can deal with. We were able to sequence all 22. We could find no obvious mutations that could explain the suppression. And so we had to use different methods. And so one method that we did, shown here on the left, is to look at the expression pattern of these 22 genes. And because the suppressor of clock is ubiquitously distributed like a clock gene, we asked how many of the 22 suppressor genes have an expression pattern that's common or similar to the clock genes. So the clock genes are shown in yellow, and the candidate genes are shown in light blue and gray in this heat map representation of gene expression. And we profiled 10 different tissues in the mouse. And what you can see is that the clock genes are clustered at the top of this heat map because their expression is high and ubiquitous. And 7 of the candidate genes, shown in blue, had expression patterns similar to clock genes and clustered with them. The other candidate genes had very different expression patterns, and that suggested that they might not be involved. So we focused on these 7 genes. They're shown here. This shows their RNA expression over time of day in either an F1 mouse, that's a suppressor strain, or a black 6 mouse. Okay? And out of these 7 genes, only one of them showed an expression difference. That's this one here, USF1, or upstream factor 1 gene. And when we looked at the levels of the PER1-2 and CRY1-2 genes, they are also elevated in the suppressor strain. Okay? When we then look at the expression of the USF1 protein, we find that there's a very subtle difference. It's a 40% increase in USF1 protein. Very small, but significant. So that focused our attention on USF1. USF1 is interesting because it's a transcription factor, and because the PER genes and CRY gene RNAs were elevated, that suggested that maybe their transcription was elevated. And so we tested the 7 candidate genes for activation of the PER1 and PER2 genes using promoter assays. And these 2 graphs show that of these 7 genes, USF1 strongly activates both PER2 and PER1 in a dose-dependent manner. But unlike clock and BMAL, which also activate PER1 and PER2, USF1 is not suppressed by cryptochrome or period, as shown here, where clock activation is suppressed by CRY1 or CRY2. This does not occur with USF1 here. So this suggests that USF1 might be acting on the same pathway as clock and BMAL. But to prove that USF1 is really the gene, we had to make a transgenic mouse, which overexpress USF1 just a little bit. This is the expression level of USF1 here in the transgenic mice. And what we found is that with this subtle increase in USF1 expression, these transgenic mice could also suppress the clock mutation as shown in these activity records on this bar graph here. So this kind of experiment shows that USF1 is capable of suppressing clock and identifies it as the suppressor of clock. So how does USF1 do this? Because we sequence the USF1 gene, there are no mutations in USF1. The only difference is the expression level of USF1. And so this suggested a regulatory mutation. And so in mice, you can do a very nice experiment. You can take an F1 mouse, an F1 of BLAC6 and BALB/c, and if the RNA has a sequence difference between those two strains, you can actually measure the BALB/c transcript and the BLAC6 transcript separately. And so we've done that in 3 different ways illustrated in this graph. And all 3 methods, I won't go through the details, show that in an F1 mouse, the BALB/c allele is preferentially increased in its expression over the BLAC6 allele. And that's really a beautiful experiment because it's the same mouse with 2 different alleles, and one allele is up and the other's not. So that shows that the regulation of USF1 is cis regulation as opposed to trans regulation. So how might that work? So to look at this further, what we did was to isolate the upstream regulatory regions of USF1. And using reporter gene assays, we could show that the BLAC6 upstream region, which is shown in blue, as compared to the BALB/c upstream region shown in green, there's a slight increase in the transcription from the BALB/c promoter. And within this region, which is about 1 kilobase of the BALB/c promoter, there are only 7 sequence differences in this regulatory region shown in green here. These 7 SNPs, or single nucleotide polymorphisms. And so we tested them, and 2 of them, SNP3 and SNP7, show a difference in transcription between the 2 strains, with the BALB/c strain being higher. And when we put SNP3 and SNP7 of BALB/c in green into the BLAC6 background, so there's only 1 difference in these 2 constructs here, we see that SNP7, shown here, can completely account for the difference between BALB/c and BLAC6. So this suggests very strongly that the difference between BLAC6 and BALB/c is due to a single regulatory change in the promoter of the USF1 gene that increases the expression of USF1 in the BALB/c mice. Okay, so how does this subtle increase in USF1 then suppress clock in a mouse? So to examine this question, what we did was to go back and try to study the binding of clock BMAL onto DNA and to see if USF1 might bind the same site. And so for a number of genes that we know are regulated by clock, called DBP, PER1, and PER2, we could find that clock BMAL bound to specific regulatory sites that are called E-boxes, which we can detect in this assay, which is called the gel mobility shift assay. Okay? And so the way this experiment works is here in wild-type mice, we have liver extract that contains many different proteins from the nuclei of liver of mice. And we bind them to an oligonucleotide that contains the E-box sequences from the DBP gene. And what you see is that we get two major bands. The top band turns out to be clock B-mel, and this lower band turns out to be USF1. So indeed, USF1 can bind to the same site as clock B-mel. And interestingly, in the clock mutant, what we see, which is shown on the right-hand side here, the clock mutant binding changes. So not only does the affinity of binding change, which I'll show you in a minute, but the nature of the binding, whether it binds as a single complex, CB1, or a double complex, also changes. So the wild-type protein binds as a double complex, CB2, shown here. The mutant protein tends to bind more as a single complex, shown here, CB1. And then on the right-hand side, when we quantitate the actual affinity of binding, what we see is in blue, the wild-type clock B-mel complex has much higher affinity than USF1, which is shown in orange and green. But interestingly, in the clock mutant, the binding affinity, which is shown in red, actually goes down to match USF1. So there are a couple things going on here. The first is that the clock mutant is actually reducing the binding affinity of clock B-mel such that the affinity is much closer to USF1. And under these conditions, we think that USF1 might be able to then compete with clock B-mel. To examine the binding of USF1 and clock B-mel in more detail, we've used a different method which allows us to look across the entire genome for where USF1 and clock B-mel might bind. And so these are UCSC genome browser views of USF1, clock, and B-mel binding to the period 1 gene. And out here you can see there's very strong binding at these sites here. Red indicates wild-type mice and blue indicates clock mutant mice. And what you can see is that in the clock mutant mice, USF1 binding is much higher than it is in wild-type, okay, to this site. This is also true for a different gene. This is DBP, shown in the second. Again, at the top here, USF1 is much higher in the mutant than in the wild-type. And over here for reverb alpha, you can also see a very big difference in these two samples here for USF1. So if we look genome-wide, what we see is that there is significant overlap in the binding in wild-type and clock mutant mice between USF and clock B-mel. But interestingly, in the mutant, the amount of binding sites go up. And in particular, if we look at this plot here, which represents USF1 binding in red, the intensity, we see that the binding intensity is much higher at sites across the genome in the clock mutant than in the wild type. But there isn't so much difference in clock or B-mel occupancy. So this suggests that indeed USF1 and clock do interact on a very large scale across the entire genome. And this could explain how USF1 is actually suppressing the effects of clock. So here's an overall summary of what we think is going on. In the BALB-C strain, there are regulatory sequence differences in the USF1 promoter that lead to an increase in USF1 expression. In wild-type mice, this doesn't really make much difference because the affinity of clock B-mel is so high that USF1 doesn't compete very well with clock B-mel. But in the clock mutant, shown here, clock tends to bind as a single complex. The affinity is lower, and so USF1 can occupy or compete for the same EBOX sequences. Now, the clock mutant protein is deficient in transcription. So USF1 actually can rescue that function because USF1 can actually activate transcription from the same regulatory sequences as clock BMAL. The only difference is, of course, USF1 is not subject to negative feedback. And in fact, that promotes the activation potential of USF1 in addition. And so we can see in this very complex genetic interaction analysis that additional factors can interact with clock BMAL at a transcriptional level, compete for binding, and actually replace the function of a mutant clock protein to rescue or suppress that behavior. So I'll stop there and acknowledge all of the people who contributed to these 3 different stories shown here. Thank you very much.